Hello everyone, today we're gonna see how to perform cell proliferation assay using Celltiter 96 aqueous 1 solution. So here now I'm just setting up the controls. So I'm using dilution assay, like dilution in the series. So based upon my cell growth, I chose my cell range of dilution between 40,000 to 800. So like how I showed in the previous video, you can look how to count the cells and dilute them and put it in the different wells so that you can compare their so that you can measure the amount of soluble formation produced by cellular reduction of MTS after the incubation. So it based on your cell growth, like how fast they grow, you can set up the range of the controls to compare. Because my cells are fast, so I did like 40,000 range to 800 cells range for well. So it's like dilution, like 40,000, 20,000, 10,000, which goes to till 312 cells per well. And so here we are using Celltiter 96 aqueous 1 solution. So that is the dark yellow one that I'm putting into each well. So I'm using 20 microliter of this solution and putting it to each well. Here I'm using the same pipette because these are my replicas and I just wanna show you in this video how to do it. And after that, after putting it into every well, even the controls that we have did, we're gonna incubate at 37 degrees for 1 to 4 hours at 5% carbon dioxide atmosphere, and then we'll come back after when it's 4 hours. And now we're gonna put our plate in a 96 well plate reader. So this is the mission. So when you click eject, you can load the plate. You have to remove the cover of the 96 well plate and just load it, and then click load on your computer. So the plate will just go inside, and you can set up the parameters. Here I have already set up the parameters. So I'm using to measure it at 490 nanometers. So the plate will be red at the absorbance from the cells at 490 nanometers, and it will give you the readings instantly. So first it just reads and shows everything in blue color, and then it later, once all the wells are red, it will give you the color change too. So based on your formation of formation, the absorbance will be varied. So by drawing the graph between the absorbance and the number of cells per well in your control, we can find the R value, and then by that we can find how many cells are there per well in our original plate, so that we can draw a graph between the proliferation among the various cell lines and how fast the cells are proliferating. So here you can see like you can compare the controls which is in the G row with the other cell replicates and draw a graph, and you can know how many cells are there. So here I'm just saving it into my folder. So this data will be given to you in an Excel format, so you can easily use Excel to calculate the number of cells that are present in a well, and then you know how many cells are present in a well. You can draw a graph for the proliferation assay. Finally, eject the plate and then close it, and you can discard the plate. Thank you.